All right, so today we're going to go over bacterial transcription and translation. First, let's go over the central dogma. This is a broad genetic and biologic principle that states that genetic information flows from DNA to RNA to protein. So the process of going from DNA to RNA is transcription, as you guys know, and RNA to protein is translation. Then you also have replication, where the DNA can create more copies of itself so that it can put a copy of the genome in each daughter cell when it's an organism divides. So this is a general guideline, but there's some exceptions. Like, for example, we have retroviruses like HIV that use reverse transcriptase. We talked about this. They make their RNA genomes back into DNA and then insert that DNA into the host genome. So, all right, so now let's talk about eukaryotic genes before we talk about prokaryotic genes. So all genes are going to have what are called promoters, which are upstream, which means just quote unquote before. So in relation to where the RNA polymerase is moving, the promoters are earlier on. So these promoters are the places at which the RNA polymerase is going to bind in order to start transcribing. And the RNA polymerase actually binds to a specific sequence called the Tata box, which is located inside the promoter. And then in eukaryotes, we also have what's called, basically we're processing RNA. So once we get the mRNA from here, you see what's called exons and introns. So these are just specific regions within the mRNA. And what's going to happen is that before the mRNA goes to become a protein, we have to edit it a little bit first. So what we do is we take out the introns and then we connect all the exons together. So these parts here are going to get cut out, and then the rest of the remaining parts are going to get all put together, and then that's the final product that'll go to the ribosome. And we don't have to talk about really why this is necessary, but just know that this process exists, and it's only in eukaryotes. This does not happen in bacteria. Okay, so genes, they also have what's called a ribosomal binding site, or an RBS. So whenever this DNA, or I guess this is already RNA, but when the DNA, let's say we have DNA, and we get the mRNA from that, there's going to be a specific site here that the ribosome, right? The ribosome needs to recognize this site to be able to bind to the mRNA, and the RBS is that specific site. And the ribosome specifically recognizes what's called the Kozak sequence, just a small region within the RBS. Okay, so let's move on to bacterial genes. These are pretty similar. They also have promoters, like we talked about, where RNA polymerase binds, but they have a specific sequence called the Pribno box instead of the Tata box. And then they also have the equivalent of the Kozak sequence in the ribosomal binding site for eukaryotes, except it's called the Shine-Dalgarno sequence in bacteria. So again, this is where the ribosome will bind to the mRNA. And then some bacterial genes are grouped together. So you have like this A, B, C, and D complex here. And whenever you have multiple genes together, it's called an operon. So these are only in bacteria. So these genes will basically all be transcribed together at the same time, making one long mRNA that can eventually be converted into, for example, here we have 4 separate proteins that are made from the operon. These sequences also have what's called a operator sequence, and we'll talk about that in the next lecture, so let's not worry about that for now. All right, so transcription, it's the same as in eukaryotes. We have 3 steps. So we have initiation. This is where RNA polymerase, the enzyme that creates the mRNA, right? It's going to get basically attracted to the DNA and bind. We have elongation. This is where the RNA polymerase is moving along the DNA, and then synthesizing a mRNA strand that slowly grows longer and longer, which is why it's called elongation. Termination, the RNA polymerase, it recognizes when it needs to stop, then it falls off of the DNA, and then the mRNA is released, and then it can go wherever it needs to go for the next process. All right, so in initiation of transcription, in bacteria, we have a specific protein that's called a sigma factor. So the sigma factor first needs to bind. So you can see this blue sigma factor binds to the promoter region first. And then the sigma factor is what, quote unquote, recruits the RNA polymerase. So the sigma factor binds first, then the RNA polymerase can recognize the sigma factor, and then it'll come. The sigma factor makes the RNA polymerase bind better to the DNA, essentially. And then for most genes, sigma factor 70 is used, just the name of the protein. And then we call this big complex with RNA polymerase plus the sigma factor. And there are a few other proteins that we don't need to discuss. We call this whole thing the RNA polymerase holoenzyme. Okay, next, elongation. So for the elongation to begin, the sigma factor is going to fall off of the DNA. Then the RNA polymerase is going to move slowly along. Well, not slowly, but it's going to move along the DNA and start making that new mRNA strand pretty much the same way that you've seen it does in eukaryotes. So nothing too big to discuss there. Then finally, termination. So this is a little bit more complicated in bacteria. So we have two methods by which termination can occur. So basically, for termination to happen, we need the RNA polymerase to come off of the DNA, and for the mRNA to also come off of the RNA polymerase, right? So first, let's look at what we call row-dependent termination. So this method, we use what's called the row helicase protein. And helicases are a class of enzymes that can basically, when you have bound two strands together, it can separate it out like this. So we use the row helicase protein. So for genes that use this method of termination, on the mRNA, there's a specific sequence called the row utilization site, or the RUT. So if you look on the right here, as this mRNA is being created, we have this place here where there's a row utilization site, right? And the row helicase protein is going to bind to that site specifically. And then as this guy is being transcribed, what the row helicase does is it slides down the new strand of mRNA, and it gets closer and closer to the RNA polymerase when transcription is done, it'll slide all the way down, and it'll basically, so at this location, right, we have the mRNA still kind of associated with the DNA. So the row helicase will unwind that and then separate the two using ATP. So basically the mRNA can now, since the row helicase has slid all the way down and unwound the mRNA, it can come off, and now that terminates the transcription process. Next, we also have, it should be row independent. That's my bad. Row independent. I'll fix that later. Row independent termination. So in this method, we don't need a row helicase, which makes sense because the name is row independent. Then the mRNA literally pulls itself off of the DNA by itself. So for genes that use this method, basically there's several regions of repeating guanines and cytosines within the mRNA. So you can see that here, right? GC-rich area. And what it does is, so the significance of that is that the guanine cytosine, right, G and C bond, G and C form 3 bonds between each other, while A and T form 2. So the bonds between are A and T / A and U. So G and C are really strong bonds. So what that means is that basically when we have these repeating G's and C's, they'll make this loop like this and then make really strong bonds here. And what that'll do is that those bonds are so strong that it'll literally rip the mRNA off of the DNA and the RNA polymerase here. So, so yeah, you can see that this is literally physically, as this loop is being made, the loop pulls off the mRNA, and then the mRNA can go and travel to the, it doesn't necessarily travel, but the mRNA can be released so that it can be translated. So hopefully that makes sense. Okay, now let's look at bacterial translation. So translation, it proceeds with the names of the steps are the same. So we have initiation. This is where the ribosome now is going to bind to the mRNA. It's going to bind to the Shine Dalgarno sequence, SD sequence. We have elongation, so now the ribosome moves along the mRNA, creating this new protein as it goes, reading those codons, getting the correct tRNAs to bring the correct amino acid, everything you should have reviewed in the video that I sent on the Google Classroom. And then finally, we have termination, where the ribosome needs to fall off the mRNA, and then the protein also needs to be released so that it can go and do whatever function it needs to do. So, like I said, the initiation begins with the ribosome binding to the Shine-Dalgarno sequence. You can see that here. So this is just the bottom part of the ribosome, and then you'd have this big component come on top as well. So you'd have the full ribosome bound to the mRNA. All right, so remember you had start and stop codons. So the ribosome is going to recognize a start codon, which is usually AUG in it's usually AUG for the majority of organisms. But sometimes in bacteria, we can also use GUG and UUG as alternate start codons. So in eukaryotes, this AUG codes for, if you look on the codon chart, if you guys remember codon charts, right, you see the 3-letter sequence and you can match it to a specific amino acid. So in eukaryotes, the start codon is basically corresponds to methionine. So the tRNA is going to bring over a methionine, and that's what starts the translation. However, in prokaryotes, the tRNA, it creates this alternate form, it carries this alternate form of methionine called N-formal methionine, and this is actually going to be the thing that initiates the elongation. So just know this. I think this popped up on a few exams, so just make sure you know this, and the fact that there are possible alternate start codons as well. Okay, third, we have termination. So again, we talked about stop codon. So the ribosome, it's going to encounter a stop codon. So stop codon here. Once it sees that, the polypeptide, so this protein that was created here, this protein is going to come off. Like you see here, the protein's going to come off, the mRNA comes off, and then along the way we use what are called release factor proteins to help facilitate that process. And now, the ribosome, it can go and find another mRNA and then repeat the whole process. All right, so interesting thing here. So this can happen in both eukaryotes and prokaryotes, but we have what's called polysomes. So this is whenever several ribosomes can attach to the same mRNA and then start reading it at once. So you can imagine, so it looks like a beads on a string, like you can see on the right. So basically, this makes the mRNA translation of the mRNA super fast because you have like 10 ribosomes all attached at the same time, and they're all making these proteins as they go. So just a term that you should be familiar with. And finally, a very important detail to know is that in bacteria, right, bacteria don't have nuclei. So normally the mRNA in eukaryotes, after it's made, it needs to travel out of the nucleus and then go find the ribosome and then do that. But in bacteria, it doesn't need to do that because there's no there's no nucleus, there's no boundary through which the mRNA has to travel before it can be translated. So that means that the mRNA can literally be translated immediately as it's being synthesized. So if you have DNA here, we have RNA polymerase, and then the RNA polymerase is making this mRNA. Even as transcription is happening, translation can occur at the same time. So the ribosome can immediately bind to this mRNA that's still being made and then start making proteins based off that. And not just one, but you'd have the polysomes here that, so multiple ribosomes can attach to the same mRNA as it's being transcribed. So this makes protein production extremely fast because the mRNA doesn't have to move at all. It's just being immediately translated. So you can see that here on the right side, you have the DNA and then these RNA polymerases, right, moving along. And then as they create this mRNA strand, these ribosomes are forming polysomes and immediately translating all of these. So you can see that on this electron micrograph here too. So this is a really key detail that they'll probably ask you about. So make sure to know this idea.